Dear students, in this lecture we are going to deal with the basics of probiotic and prebiotic foods. As you all know that microorganisms are found everywhere on earth. They are commonly known for their role in causing disease, but their power has also been exploited to the benefit of humankind. As we all aware that the microbes are used for the production of varieties of fermented foods like pickled vegetables, wine and beer, cheese, yogurt, etc. They also impart a selective role in the production of drugs such as antibiotics and food ingredients such as vitamins and citric acid. Most of the gut bacteria are present in the large intestine. The usual residents of the gut interact with their host in a very difficult manner. They modulate the effect of potentially harmful bacteria, have an impact on the host GI tract, digestion, metabolism and immune system, and also influence functions beyond the gut. On completion of this lecture, you will be able to infer the role of prebiotics and probiotics in human health, and also able to identify the different sources of probiotic strains and prebiotic foods. Before moving into the probiotic foods, you should know about the history of probiotics. The term probiotic is derived from Greek, which means for life. Regarding the origin of probiotic foods, the effect of probiotic began with the consumption of fermented foods, which was observed in ancient Greek and Romans. It was witnessed that the Bulgarians were healthy and long lived because of their regular consumption of fermented milk products, which consisted of lactobacillus species. These bacteria supported the growth of good microflora in the gut and decreased the microbial toxic activity. The term probiotic was first used by Parker in 1974. Regarding the definitions of probiotics, many scientists have framed the definitions and its positive effect on human health. Fuller in 1989 defined probiotics as a live microbial supplement which affects host health positively by improving its intestinal microbial balance. Naidu et al. In 1999 described probiotic as a microbial dietary adjuvant that beneficially affect the host physiology by modulating mucosal and systematic immunity as well as improving nutritional and microbial balance in the tract. In 2001, Skrisenmeyer and Devers stated probiotic as a preparation of a product containing viable defined microorganism in sufficient numbers which alter the microflora in a compartment of the host and by the exerting a beneficial health effect in the host. Later on 2001, the Food and Agricultural Organization and World Health Organization defined probiotics as live microorganism which when administered in adequate amount confer a health benefit to the host. Now let us see the need for probiotics. Probiotics are a live microorganism similar to beneficial microorganism naturally found in the human gut. These good bacteria are used to prevent and alleviate many conditions, particularly those that affect the gastrointestinal tract. These good bacteria in human gut facilitate digestion, absorb and provide nutrients such as vitamin B, K, folate and short chain fatty acids. Around 10% of an individual daily energy requirement is the byproducts of beneficial gut bacteria. These good microfloras develop under natural conditions and do not need any bacterial supplement. But due to the changing lifestyle and food habits affects the gut microflora and its colonization. For example, intake of antibiotics and other drugs, microbial infections, consumptions of low fiber food, chronic diarrhea and stress are some of the factors affecting the intestinal micro ecosystem. Any imbalance in the gut microflora results in physical discomfort, susceptibility to infection, cancer, aging, reduced efficacy of medications, radiation, chemotherapy, poor nutritional response, chlorinated water and deficient immune response. Therefore, food supplemented with probiotics helps to recover gut imbalance and thus provide favorable effects. Next, we will see about the sources of probiotic strains. The most commonly used probiotic organisms generally belong to the genera Lactobacillus, Lactococcus species and Bifidobacterium. These organisms have been recognized under GRAS, that is, generally recognized as safe. Other microbial species besides lactic acid bacteria, Bacillus subtilis, Propionibacterium species and Saccharomyces boulardii have been recognized as source of probiotics. Probiotic food supplements may consist of single strain or mixed cultures of two or even more strains. Lactobacillus acidophilus, Lactobacillus casei, Bifidobacterium bifidum are some of the commercial strains of probiotics. At this juncture, it is very important to know about the mechanism of action of probiotics. The mechanism of action of any probiotic food supplement, it depends on their metabolic end product such as organic acid, which lowers the pH in the human gut and subsequently inhibits the growth of pathogenic microbes. The major action mechanism includes the enhancement of epithelial barrier, increased adhesion to intestinal mucosa, associated inhibition of pathogen adhesion, competitive exclusion of pathogenic microorganism, production of antimicrobial substance and modulation of the immune system. For probiotic to be highly effective, the following attributes are necessary. It should assure shelf life such that they will contain adequate number of viable organisms at the time of ingestion. It should provide safety for human consumption. It should possess ability during processing, storage and transportation. It should not displace the native bacteria already present in the gut. It should have the ability to survive in the upper digestive tract. It should have high resistance against acid and bile environment. It should have the ability to survive and grow in the intestine. It should produce antimicrobial substance like bacteriocins. Also possess the ability of adherence to human intestinal cell lines. It should be able to colonize in the gut and produce favorable response in the host without any toxic adverse effect. Let us now focus on the effects of probiotics on human health. Intestinal infection diseases caused by pathogenic organisms are the major cause for death in most of the countries. The intake of probiotic supplemented food confers health benefits to the host, and different degree of evidence also supported the benefits of probiotic foods. The effect of probiotics on human health includes manages lactose intolerance, improves immune system, reduces the risk of colon cancer, reduces cholesterol level, lowers the blood pressure, reduces inflammation and allergic symptoms, beneficial effects on mineral metabolism, particularly bone density and stability. It also reduces the pathogenic microorganisms, prevention of osteoporosis, prevention of urogenital infections. It also suppresses the endogenous pathogens and control the irritable and inflammatory bowel syndrome. From the previous works, the effect of probiotic food in the production of beta-D-galactosidase have been observed. Lactose intolerance in few people is due to the inability to digest lactose present in milk. This inability is due to the absence of beta-D-galactosidase in the human intestine, which leads to the abdominal discomfort. It was found from the studies that specific bacteria such as Lactobacillus acidophilus and Bifidobacterium bifidum were able to produce enzyme beta-D-galactosidase, which is responsible for hydrolysis of lactose. The consumption of food supplemented with such specific bacteria improved the tolerance of dairy products. These microorganisms are proved to have antimicrobial properties through the production of substances such as organic acid, hydrogen peroxide and bacteriocins, which helps to control the intestinal infections. Several animal studies reported that the consumption of fermented dairy products supplemented with probiotic bacteria inhibited tumor formation. Regarding the list of probiotic foods, the most common probiotic foods are fermented milk products such as yogurt and fermented dairy beverage. The highest challenge in manufacturing probiotic food is incorporating a live probiotic microorganism into foods and keeping them alive throughout the shelf life. The ingestion of probiotic food with a dosage of about 10 to the power 8 to 10 to the power 9 viable cells per day attributes to healthy effects. Therefore, International Dairy Federation suggested that foods containing probiotic organisms need to have more than 10 to the power 6 to 10 to the power 7 colony forming unit per gram at the time of consumption. The viable count of probiotic organism generally reduced to 10 to 100 folds during storage. Hence, desirable viable count can be achieved by introducing high number of probiotics during manufacture. The consumption of probiotic organisms at high dose does not appear to pose a health risk. Let us discuss about the points to be considered while manufacturing probiotic foods. Selection of well-suited combination of probiotic strain and type of food. The food processing condition should be compatible with the probiotic survival. For fermented foods, food matrix should support probiotic growth during fermentation. The selection of product matrix, packaging material and environmental condition should guarantee acceptable probiotic survival during product supply chain and storage. The addition of probiotic does not adversely affect the taste and texture of the food products. Probiotics can be integrated into foods and beverages as dry powder blended into foods such as infant foods, dispersed into liquid or semi-liquid products such as juice or ice cream, and also inoculated into fermented products such as yogurt and fermented milks. The commonly available probiotic foods are yogurt, kefir, miso, natto, and most vegetables that are fermented have probiotic values. Other probiotic foods include ice cream, margarine, non-fermented fruit and berry juices. Probiotic muesli, nutritive bars and breads are found in world market. Let us now move to the next topic, prebiotics. Regarding the history and origin of prebiotics, it was first recognized by Japanese in the form of non-digestible oligosaccharides. They are also recognized the significance of oligosaccharides in human milk and later confirmed that the consumption of fructo-oligosaccharides and galacto-oligosaccharides lead to an increase in intestinal bifidobacterium and stimulated their growth in the human gut. The term prebiotic was coined by Gibson in 1995. Prebiotics can overcome the problem associated with probiotics. Few bacteria such as bifidobacterium require very specific nutrient needs through which their population increases. Research has revealed that when prebiotic foods are consumed, the quantity of specific bacteria increased up to 100-fold. But also noted that when intake of prebiotic was stopped, the number of gut returned to their original values. This proved that instead of giving an external bacteria into the gut, the number of resident bacteria can be increased. As per the scientist Gibson, prebiotic is defined as a non-digestive food ingredient that beneficially affects the host by selectively stimulating the growth and activity of one or more limited number of bacteria in the colon and thus improves the host health. The International Scientific Association of Probiotic and Prebiotic defined dietary prebiotic as a selectively fermented ingredient that results in specific changes in the consumption and or activity of the gastrointestinal microbiota, thus conferring human health. Why this prebiotic is needed for us? Let us discuss the reason. Prebiotics are non-digestible food ingredients that selectively stimulate the proliferation and or activity of desirable microbial population already resident in the consumer intestinal tract. In more common terms, prebiotics are foods for bacterial species which are considered beneficial for health and well-being, and it is scientifically accepted. Prebiotics are valuable dietary additions for modulating the growth and activity of specific bacterial species in the colon that are considered health supporting. The prebiotic carbohydrates are not digested by human enzymes but are fermented by the flora of large intestine. Thus, they increase the biomass, feces weight and feces frequency, have a positive effect on constipation and on the health of mucosa of large intestine. Regarding the sources of prebiotics, carbohydrates are the well identified source of prebiotics. The prebiotic attributes are found in most of the oligosaccharides. They are carbohydrates with similar to that of dietary fibers are found in form of fructans in vegetable as well as in the milk of mammals. The major prebiotic components are fructans or fructo-oligosaccharides, polyfructan, inulin, galacto-oligosaccharides, lactulose, lactose, sucrose, polydextrose, arabinoxylans, cereal fibers and resistant starches, as well as some polyol structures, lacticol and isomalt, etc. The natural sources of prebiotics occurs in foods such as chicory, cereals, agave, whole grains, banana, root vegetables, marine algae and milk. Most of the commercial prebiotics are synthesized by chemical or enzymatic methods. Let us see how this prebiotics works. The main action of prebiotics occurs in the colon. The prebiotics delivers their benefits through the enhancement of growth and or the metabolic activities of bacteria that ferments them. The primary target for prebiotic action is Bifidobacterium and Lactobacilli. The action of prebiotic is indirect and it causes the selective modification of the host microflora. It also induces changes in the microflora which is further responsible for the beneficial effects. The anti-lipemic effects have been associated to decreased cholesterol absorption, increased excretion of cholesterol through feces and by the production of short chain fatty acid such as acetate, butyrate and propionate produced by intestinal microflora fermentation. Any foods to be prebiotic, it should possess the qualities such as it must be non-digestible or sometimes partially digestible. It should possess the resistance against the effects of gastric acidity and digestive enzymes. It should be non-absorbable in the small intestine and it should not undergo hydrolysis or absorption in gastrointestinal tract, and it should not be fermented by pathogenic bacteria in gut. It must be fermented by intestinal microflora. It should possess selective stimulation of the host own beneficial microbiota. In relation to the human health, the prebiotic food offers many advantages like it improves bowel function, it increases stool frequency, it increases the production of short chain fatty acid, improves the bioavailability of minerals, mainly calcium and magnesium. It promotes the growth of health promoting bacteria such as Lactobacilli and Bifidobacter. It can reduce insulin concentrations and lower triglyceride levels. It helps in the management of infectious diarrhea. It possesses anti-carcinogenic effects on human health and also reduces the risk of cancer. Few studies on rats and mice showed a decrease in fat mass development due to feeding of fructans. Other fermentable dietary fibers such as pectin and beta-glucan oat bran have been shown to lower serum cholesterol in rats. Similarly, a decrease in total serum cholesterol level was observed after supplemented with inulin in mice and rats. Even the pet food and animal feed were added with prebiotics, which improves the control of gastrointestinal infection and enhancement of growth performance was particularly observed in poultry. Most of the commercial prebiotics are oligosaccharides and dietary fibers. Foods with prebiotics are found in commercial market in form of dairy product, infant foods, bread, fruit juices, margarine, pasta, dairy desserts, cereals, yogurt, biscuit, soft drinks, in general, isotonic drinks, liquid sugar and modified sugar, chocolates and candies. The added prebiotics are mostly acquired either by extraction from plants, by enzymatic hydrolysis or by synthesis from mono or disaccharides such as sucrose or lactose. A normal diet provides 5 and 10 grams of non-digestible carbohydrates per day. This includes oligosaccharides from vegetable origin, especially fructooligosaccharides. Effective doses of oligosaccharides are in the range of 5 to 10 grams per day for healthy adults. Doses less than 5 grams are generally considered ineffective. Once if you are aware about the probiotic and prebiotic foods, now it's time for you to understand a new term called synbiotics. Synbiotics are defined as a mixture of probiotic and prebiotic that beneficially affect the host by improving the survival and implantation of live microbial dietary supplements in the gastrointestinal tract by selectively stimulating the growth and by activating the metabolism of one or more limited number of health promoting bacteria, thus improving the host health. In simpler terms, they are the mixture of microbial and non-microbial agents. As discussed earlier, probiotic bacteria colonize in the upper part of the intestine to avoid the adhering of pathogens to the intestinal tract and help in digestion. A prebiotic is a fiber such as fructose oligosaccharide, galactose oligosaccharide, etc., and it is intended to stimulate the microflora in the large intestine. The combination thus works separately in the small and large intestine, but synergistically as they increase the overall gut health. Synbiotic foods have the ability to reconcile and normalize the intestinal flora, particularly after the destruction of microorganisms following antibiotic, chemotherapy or radiation therapies. They are also capable of suppressing the development of putrefying process in the stomach and intestine, thus preventing the incidence of numerous diseases such as food allergies, ulcerous colitis, constipation, diarrhea, cancer and gastrointestinal infections. A symbiotic product contains 500 million to 5 billion live microflora can be taken several times a week. A few examples of symbiotic food combinations that can be made to achieve the effect of both probiotics and prebiotics are yogurt or kefir and honey and acacia gum, legumes and pickles, yogurt with oats, kombucha tea with acacia gum, yogurt or kefir with acacia gum and honey, sour pickled onions and pickled garlics. Finally, we summarize the points so far learnt. Probiotics are live microorganisms similar to beneficial microorganisms naturally found in the human gut. The most commonly used probiotic organisms generally belong to the genera Lactobacillus, Lactococcus and Bifidobacterium. The intake of probiotic supplemented food confers health benefits to the host, and the different degree of evidence also supported the benefit of probiotic foods. Prebiotics are non-digestible food ingredients that selectively stimulate the activity of desirable bacterial population already present in the consumer's intestinal tract. The prebiotic attributes are found in most of the oligosaccharides. The prebiotic delivers their benefits through the enhancement and growth of metabolic activities of bacteria that ferment them. Synbiotics are the mixture of microbial and non-microbial agents. With this, we complete the topic related to fermentation of foods and its allied products. Thank you.